now. The good news of waiting is that even more therapies came out during that time period. So I put in quotations here, new COVID therapies, because frankly, COVID is new in itself. So all of our therapies for it are somewhat new, but I'm afraid by the end of my talk, given the speed of how much things are changing, that these might be old therapies. So let's get right into it. For those that don't know me, I'm an infectious disease physician up here at Morgantown at WVU, and I am the program director for our fellowship here. So I'm going to start talking first about our novel SARS-CoV-2 monoclonal antibodies that just got approved in the last month. So the first one is a drug called Bamlanivimab, or I call it the Lilly product. It's a little bit easier to say. It had its EUA granted on November 9th of this year, and it is a monoclonal antibody. So this is an antibody that is a fully human monoclonal antibody that's directed against the spike protein of SARS-CoV-2. And so what that does is it allows it to block the attachment of that virus to our cells, and therefore block the entry of this virus into our cells. And I'm going to have a little bit of a schematic and another slide or two that shows how that works. In the EUA, though, it was approved only for outpatient use at this time in patients with mild COVID-19 who are not hypoxic, so not requiring any oxygen support, and really in that group who have risk factors for progression to severe disease. And for instance, some of those risk factors would be an age over 65, obesity, and I'm going to get into a list of other comorbidities that were supported based on one trial we have so far with this drug. It was also looked at in hospitalized patients, but the clinical trials in hospitalized patients were actually stopped due to no benefit seen among placebo versus study drug and a concern of risk of adverse reactions. The second agent we'll talk about is the imdevimab, which is the Regeneron product. And this is a combination of two different monoclonal antibodies that again target that same spike protein of the SARS-CoV-2 virus. And this one is very similar to the Bamlanivimab in that there was some concern in those who were hospitalized and on high oxygen that this was not going to be a product that would be beneficial. They saw some safety concerns. And so there was an outpatient trial data that really showed success with a reduction in both viral load, time to recovery, and medical visits for COVID-19 specifically following the infusion. And I'm going to get into details of those trials as we go through this talk. But it was just released for an EUA on November 21st, 2020. So within a matter of 2 weeks, we had two new monoclonal antibodies that were released under the EUA. So this is a sample of what Bamlanivimab does. And so you see our host cell here, which has the ACE2 receptor, and that's really the target site for SARS-CoV-2 to enter our cells. And remember, this ACE2 is found both in pneumocytes, so those cells in our lungs, as well as in some of our other organs like the GI tract and the goblet cells in our secretory passage. And this is what the virus itself looks like, a schematic of that. And that red protein there is the spike protein. And again, without having that spike protein open to bind, the virus could not bind to our ACE2 receptor or enter the cell. And so this is where Bamlanivimab comes in. It comes in, blocks that spike protein from ability to bind, and therefore moves the virus along so that it cannot enter our host cell. And so this is, again, a fully human monoclonal antibody. It's a single dose of 700 milligrams one time. And the side effects that were seen in the BLAZE-1 trial, which is the randomized control trial that supported its release, was that there was some mild side effects seen with nausea, vomiting, and headache, as well as some infusion reactions. There was both one episode of anaphylaxis and one serious infusion-related reaction in the treatment arm. Both of those resolved. Patients actually completed the serious infusion-related reaction. They did complete the infusion, whereas the anaphylaxis, obviously, it was stopped. It was reported in about 2.3% of patients in the group had any type of infusion-related reaction, compared to 1.4% in the placebo. So certainly higher than placebo, so something to be concerned with and to watch, but overall quite safe, it appeared. And there wasn't identified in that study any drug interactions or disease state interactions that would be a contraindication to its use. And these were the studies to date. So the ACTIV-3 trial was the inpatient study, and that's a part of the NIH group of active trials. And there was insufficient evidence that the use of Bamlanivimab in our inpatients improved outcomes, and so that was actually stopped. ACTIV-3 is ongoing, but with other study drug agents and not with this agent. The BLAZE-1 trial is really where we saw the benefit. Again, this was outpatients, mild to moderate disease, and the key here was the timing, within 3 days of their test result. And on average, it was about 4 days from symptom onset. So these are in a study designed where you can get a rapid test result, a rapid entry, and rapid use of the study drug. And what they saw in their outcomes was with hospitalization rates, it was a decrease of 1.6% in everyone who received study drug versus 6.3% in the treatment arm, in the placebo arm. So a reduction of about 5% in hospitalization rates overall. When they looked into subgroups and they wanted to see, okay, what about those patients that we know are at risk for severe COVID infection? So we think our older population, those with obesity have been identified. And so in those two different groups, the hospitalization rates were dropped from 15% in placebo arm to 4% in the treatment arm. And so that's really what got the benefit for the EUA to be released and to be released in that population of high-risk groups. There's a third trial with this drug going on right now. That's the BLAZE-2, and that's in long-term care residents and staff. But this is more of a prophylaxis trial where if we administer before someone becomes infected in a setting of where there is an active case, could we prevent disease? And so that's ongoing. We haven't yet seen the results released from that trial. So with that, the EUA came out, and this was the guidance that came out. So high-risk COVID-positive outpatients who had at least one symptom, so not using it in asymptomatic patients, who are being seen within 10 days of their symptom onset. And so this does ask us to hopefully have a fast turnaround in terms of identifying a patient who would be at risk for severe disease, have a positive test, who can get in early. And these were the risk factors they identified. So older age of 65, a BMI of over 35, chronic kidney disease, diabetes, or other immunosuppressive disease or on immunosuppressive treatment. Their second group was those who were 55 years of age and older who had risk factors of heart disease, hypertension, or lung disease, chronic lung disease. And separately, there was a look at children ages 12 to 17 with specific risk factors. Some of those risk factors included sickle cell disease, congenital heart disease, but other kind of immunosuppressing situations, as well as severe obesity in children. And again, it's a one-time infusion. The caveat to this is that you do need to monitor for that infusion reaction. So it takes about an hour for this to run through, and they recommend at least in the EUA that you monitor for a full hour after the infusion completes. In some cases, or in some of the study, they follow them out for 2 hours after the infusion completed, but by the EUA guidance, it's at least 1 hour. So what are the concerns that some of us might have with this drug? It's limited published evidence to date. So the N of the group that got study drug was about 300. And so I think this is common for COVID studies right now. We want to get drugs that might be beneficial out as fast as we can. So we're not going to have that robust numbers to look at. We don't really know who will benefit the most from that one trial. It looked like older people with risk factors. And again, when is the optimal time to administer drug? At least from this study, it looked like it would be within that 4-day of symptom onset, or at least within a few days of that positive test. If we think about how this drug works, it does sound like we should use it as early as possible. If I want to prevent the virus from entering the cell, I want to do that as early in the infection as I can. How do we allocate a limited supply, particularly upfront right now with not having a lot of supply for the whole state? We know that within our own state, we have a large population of elderly patients who do have risk factors. So if we go just by the EUA, we're going to have a wide open group of people who would meet criteria. And then you have to have the logistics for this. You need an infusion site where patients who are infective, right, so they can infect others, so they can come in safely, sit for that 2-hour period at least, if not longer, to get their infusion. And you need the staff and the workflow for that. So that's something that, unfortunately, we don't all have readily available in our sites. It's not as simple as coming into the clinic for an injection. We certainly don't want to use our cancer infusion sites with actively patients getting chemo with patients who are coming in COVID positive. So we have to figure out where we can split people up and so forth. I'm going to quickly go to the Regeneron product, which is, again, that combination monoclonal antibody. And as you can see from this diagram, this has 2 separate monoclonal antibodies that target the spike protein. And they do it in a non-competing way, which means you're going to have 2 active drugs blocking that spike protein, which is the theory there would be the hope that it would have even more success than a single monoclonal antibody. And this is, again, another single infusion of 2,400 milligrams. It's 1,200 of each monoclonal antibody into one single run. And the side effects that were reported in this one, so they classified serious adverse events, 1.6% or 4 people in the 2,400 milligram dose compared to 2.3% or 6 people in the placebo group and less than 1% in the 8,000 milligram dose. And none of those serious adverse events that were reported were related to study drug. Examples of those events were people who had nausea, vomiting, developed pneumonia, developed hypoxia, which was felt not to be related to the drug itself and more related to the disease process. If we look at infusion reactions or hypersensitivity, There was a higher proportion seen in the larger dose, the 8,000 milligram dose, with 0 seen in the 2,400 milligram dose. So in those small numbers, at least a little bit better than the Bamlanivimab product. And then the other thing they compared was that high dose or low dose. Was there any difference in outcomes in their study? And their study was looking at viral load reduction, which we could argue, what does it mean if the viral load is less? We do know that virus can be detected for quite some time without it actually being replicative or infectious. But it also showed no difference in medical visits after the infusion for COVID-19. And so with that, the EUA was approved for the lower dose because of a less risk of infusion reactions with the same benefit seen. And these are the studies with the Regeneron product here. The first 3 here are drug-sponsored trials. And so this was the outpatient one, which is really what got the EUA. They had a total of almost 800 patients split into 3 groups, one with placebo, one with 2,400 milligram dose, and one with 8,000 milligram dose. And so what they saw, if they looked at study drug group, which is 434 between those 2, versus 231 in the placebo, the total 28-day hospitalization rate went from 2% to 4%, or from 4% to 2%. So there was a reduction in treatment group. When they looked in the high-risk group, they saw a better benefit, a reduction from 9% hospitalization rate to 3% hospitalization rate. So, similar reduction that we saw in the prior drug. Small numbers, so make note that that is a small number of only an N of 78 and 150. This is ongoing, so we will get better data as they are able to enroll more people. Their inpatient study also compared different cohorts of oxygen support. And so they had simple nasal cannula to no oxygen support, and then to our higher levels of support. And they haven't released the data on that yet, but they did actually temporarily stop enrollment into the high oxygen requirement groups, again, for lack of benefit and concern for safety. Their third trial is similar to what we saw with the Bamlanivimab in the nursing home, but this was in households and seeing about prophylaxis with household contacts of COVID patients. So if I administer this to those who are in the same house with someone who did test positive, before they test positive, can I block their acquisition of disease, or at least their severity of disease? And that's still enrolling. They've been enrolling since July, but we have not yet seen the data from that trial. And then the recovery trial, so if those of you who remember, the recovery trial is the trial that's in the UK, that really where the dexamethasone support came from. And so they are including this monoclonal antibody cocktail, and they're hoping to get 2,000 patients enrolled into that study arm, where this versus standard of care. It's pending. They actually did not stop enrolling in the high oxygen group. They reviewed their own safety data after the pause here, and have continued enrolling into both low oxygen support and high oxygen support. But again, we haven't seen the results from that yet. So this is what the EUA came out with, and I'm just going to show you and not read through everything again. It's the exact same guidance as what we saw with the Bamlanivimab product. So it's the same high-risk groups that they're targeting, the same time set within 10 days of symptom onset is the hope of when you would get that person study drug. So what are our concerns? If the risk factors are the same and the data is basically the same, the concerns are going to be the same. So there's, again, limited published evidence to date. So in that study, only 500 received study drug, the rest was placebo. Who will benefit most? It appears to be the high-risk group, which would make sense, as we know they're more at risk for hospitalization. How do we allocate our limited supply? And how do we identify of those who would qualify as high risk to really be the ones most beneficial for getting this drug? And then the same issue from a logistics standpoint. So those are the 2 new monoclonal antibodies that were released just in November through EUAs. Another one that was released, which hasn't gotten as much, I think, publicity that we may not know about, is Baracitinib, which it had its EUA granted on November 19th. And this is a different study population. So this is for hospitalized patients who are hypoxic. So they are receiving either nasal cannula, high flow, on the ventilator, or ECMO. And this was in combination with remdesivir. So the EUA specifically states that you have to use both in its use. And so this is a Janus kinase, or a JAK inhibitor. And so the goal of the JAK inhibitors are really to try and block that inflammatory pathway, or that kind of cytokine pathway that can make really autoimmune or inflammatory conditions worse. And so this is not a drug that was developed for COVID. This is one that's already been approved actually for rheumatoid arthritis and in use, but looking to see, can we repurpose it in those patients who get into that cytokine storm pathway. And the data for this came from the NIAID Act 2 trial. Again, another randomized double-blind placebo-controlled trial. And they had 2 arms, about 500 patients in each, and they were looking at time to recovery within a 29-day period. And so in the patients in the study arm, they got both Baracitinib and remdesivir versus placebo remdesivir. And What they found was that the median recovery time, defined as either discharged or still hospitalized but not on oxygen support, was reduced, very modest, 7 days versus 8 days between the two groups. So not a robust difference there, but there was a little bit of benefit seen. And so therefore there was an EUA that was released for that. This is not a drug that is being allocated by the federal government to states individually. And so this is one that would be already available. You could order if you wanted to order it in certain patients, but again, it's not getting as much support as the other ones. And the reason for that are a couple of concerns. Is, again, it's not a specific SARS-CoV-2 targeted drug. So we try to use these immune modulators that act on parts of the cytokine pathway. We may block one thing, but we're not going to be able to block the whole pathway. And so the benefit of that might not be as robust as something that targets specifically the virus itself. It's also more far-reaching in the fact that it's immunosuppressive activity. And so we already know that some of our COVID patients, particularly those with multiple comorbidities who are at high risk for hospitalization, we place them on high-dose steroids that we're concerned for infectious complication arising as their disease progresses. We've seen a few cases here of invasive aspergillus pneumonia that's been reported in other places as well. So if on top of that I'm giving them an immunosuppressive agent, I need to be on the lookout for that. And the differences in time to recovery was quite modest compared to placebo of 1 day. So this is a drug that I think there is, a theoretical benefit there, but what is the risk of complications, the cost of drug as to when we might use it? So I'm going to summarize so that we have time for questions. And so within 1 month time period, we had 3 new EUAs for therapies against COVID-19. And these are, again, small number trials with ongoing recruitment, which may lead to actually different outcomes and findings. And so I say this to think about remdesivir. So we've had more study results that have come through with remdesivir that really show the benefit might be in specific subgroups of patients with COVID-19. The WHO Solidarity Trial just published their preliminary data, I think today or yesterday, in the New England Journal of Medicine. And though they did not see a benefit in mortality, which we already knew with remdesivir, they did not see a benefit of time to recovery. But that's a different trial. It's across different countries. You could not, it wasn't a placebo-controlled trial. It was comparing different remdesivir to other therapeutic agents. But they did not literally release their subgroup analysis or their time from symptom onset. So again, as more data comes out, we might find where these drugs work better. From the NIH trial with remdesivir, the benefit was seen actually in those on supplemental O2 only, with no benefit seen in those on ventilator or ECMO or high flow. And so we may see from our monoclonal antibody trials more data coming through with more numbers. And then there's the aspect of the real world outcomes that can be different. So these monoclonal antibodies were given within 3 days of a diagnosis, and again, on average within 4 days of symptom onset. If I think about our patients here who get tested, they may wait till day 2 or day 3 of symptom onset to get tested. And then if their test result takes 3 to 4 days to come back, we're looking kind of outside that window of where we may have a beneficial from this therapy. And so we need to really figure out how can we get a faster turnaround on our testing in order to get this drug used. And lastly, I think this just points to the need for, again, ongoing clinical trials to identify more therapeutic agents for COVID-19. We're in the midst of a huge surge countrywide, but certainly we've seen it in our own state, in our own hospitals. So we need more research to identify other medications that might help in outcomes here, because obviously one drug's not going to be the answer for our patients. So I'm going to stop there so that we have time for questions.
Thank you so much, Dr. Reese. That was excellent and lots of great information there. We did get a couple questions in the chat, but before we jump right into that, I wanted to ask any of the other hub members on the call, if there was any comments you wanted to make, feel free to unmute as well.
This is Lisa Costello. I just wanted to take this opportunity to say thank you to Dr. Reese. So the state has compiled kind of work groups to look at all these new therapies, and Dr. Reese serves on all of them. And I think that it's really helpful to have input from individuals on the ground. The workforce is comprised of individuals from across the state. So I thought Dr. Reese did a great job giving an overview, but wanted to thank her for all of her efforts in serving in those roles. I don't think publicly people know that there's that happening behind the scenes, but it's a lot of extra time, and there's others on this meeting that are involved too. So just want to express public appreciation to their great efforts.
Thank you so much, Dr. Costello, and I'm happy you could join us today as well. All right, so we'll just jump right into the Q&A. So the first question we got, and I do apologize in advance for my mispronunciation, is Bamlanivimab in an outpatient setting?
Yes, so the approval for it is only to be used in an outpatient setting for patients who are not hospitalized yet with COVID-19. Because the goal of it and the benefit that it showed was that it would prevent hospitalizations. So to use it in a hospital, you're already beyond where its benefit would be.
Excellent, thank you. Any other comments from any of the other hub members? All right. The second question we got was, do you know of a trial that used monoclonal antibodies with remdesivir?
Well, so I think the trials that were halted, so those inpatient trials using both Bamlanivimab and the Regeneron product did include remdesivir, where that was a standard of care. And so in those trials, again, they weren't seeing benefit among the hospitalized patients. And so though some are still recruiting, others were halted.
Great, thank you so much.
I will say it does beg a question. So if we think about the activity of remdesivir being an antiviral, we're only looking at that in hospitalized patients because it's an IV medication. We haven't used that or thought about that. If I gave a dose at an outpatient, would it actually have a better benefit in that early phase? And so that's not a study that's been done, given that is an IV antiviral product that's in the hospitals only.
Thank you, Dr. Reese. Another question we got in the chat was, do you have any information of convalescent plasma and monoclonal antibody combination therapies?
So I would have to go back to the studies. I don't believe again, the benefit was being seen only in outpatients. Those outpatients were not getting convalescent plasma. I would say in those inpatient trials where convalescent plasma, like on the recovery trial where convalescent plasma is considered, could be standard of care, they might be getting both of those, but we don't have that data yet.
Great, thank you so much. And another question we got was asking if there's any comment on outpatient use of dexamethasone.
So I think I've heard, not really through the grapevine, but in some places people are considering it. I think if we look at the data of it and from the NIH guidelines for COVID-19 treatment, it does recommend using dexamethasone in those who are hypoxic. So in the hospital on oxygen, they actually don't even recommend starting it in patients not on oxygen yet in the hospital. And so I think from the recovery trial, the benefit was seen in hospitalized patients. I don't know that we can strategize it out to outpatients yet. But I welcome anyone else's comments on the steroid aspect too.
Yeah, no, I would agree with everything you said. There is precedent for other coronaviruses, particularly the Middle Eastern respiratory virus, where actually dexamethasone increased the time to viral clearance. And so, I think that is sort of the other side of the scale, weighing about why you might not want to do that if you don't really need it.
Thank you all. So another question we just got was there a cutoff on upper end of BMI in the trials? I noticed that BMI greater than 35 was used as an inclusion.
So there wasn't a cutoff for a BMI that I can recall from the trial, and 35 wasn't the cutoff for the inclusion in the trial. It was just within the trial where they saw the benefit was best. It was in those who had a BMI of 35 or over, but they actually had people who were under 35 in that trial. The EUA specifies that BMI of 35 or higher should be a risk factor that would qualify them for it.
Great, thank you so much. So we don't have any other questions coming in the chat as of now. Dr. Hodder?
Well, no, I was just going to ask Becky or Alisa if they would like to comment on what has been established for the state guidelines for Bamlanivimab.
Yeah, sure. Lisa, I'll let you finish after I speak because it might have changed since yesterday. We actually just met again yesterday as a group to change our initial recommendations. So I talked about, within these slide sets that our concern is we've gotten a certain allotment, and do we want to run through that too quickly by opening it up for everyone who would meet the EUA? And so we had initially recommended that the state use would be for those 65 and older with 2 additional risk factors because we wanted to try and make our supply last. But then we're left wondering the people that we're missing, if that's too strict, if we're not using enough of it now. There is a time from a logistics standpoint to get into it. So yesterday we met and we were changing the guidelines to say, okay, you need 2 risk factors, 65 being one of them, or you can be have BMI of 35 and chronic kidney disease, or diabetes and a BMI of 35. So we're trying to open it up a little bit to get the product out there. I don't know if that's been fully released yet, though, so I'll go to Lisa on that.
So thanks, Dr. Reese. So yes, we met with our monoclonal antibody therapy work group, and what Dr. Reese just described was accepted by pandemic leadership. So it will open up the criteria in that you have to have 2 of those risk factors. We added in the exclusion criteria being less than age 55 for the reasons I think Dr. Reese probably touched on in regards to where we feel the drug to be of most benefit. But the thought was is that we didn't want to be so restrictive while we have product available in our state. And we changed the name, so it's no longer just the Bamlanivimab, which Mitra, I also can't say it. So, I mean, we just, a lot of people refer to it as the Lilly product, but we're now calling it our monoclonal antibody utilization, because as we get Regeneron into our state, which should be very soon, the criteria will be applied the same to that product as well. So it will be, as Dr. Reese said. Interestingly, in the pediatric population, you know, it was discussed age 12 through 17 with a BMI percentile over the 85th percentile. And so our state hasn't developed a separate pediatric criteria for use, and it is more so going to take a on basis, you know, evaluation by whether it's pediatricians or pediatric infectious disease doctors who felt that may be warranted, because we feel that it would likely be a small population, perhaps people living with cystic fibrosis or other higher risk conditions, and would still obviously have to meet criteria within the EUA. But so that's where we're at from a state level. There should be correspondence that individuals will be receiving through various channels that kind of further explain that, and certainly we want providers to know that this treatment is available out there, and then provide resources on how they could find it if they have a patient who is necessary. We've been hearing from some of the centers that did receive the initial supply of Bamlanivimab that they're just not identifying patients to give it to. So I think some of that comes with provider education. What better way than Project Echo? But to, you know, let people know that this treatment is out there, as Dr. Reese just nicely presented the data of where we have seemed to find it thus far to be most beneficial. And if in your individual practices you identify potential patients, you know, working with the different resources that the state have made available as to where you can get that medicine for your patient. Because as Dr. Reese said, there are logistical challenges in the infusion. We've worked a lot. I didn't see Dr. Krupika on the phone, but Dr. Krupika has worked a lot in regards to the nursing home population and ensuring that the drug can be mixed, if you will, and then sent to a nursing home so that the infusion can be performed there, so that, you know, hopefully we can keep some of that higher risk population out of the hospital as well. And so I think there certainly have been logistical challenges with the administration of these monoclonal antibodies and the need to be able to monitor for any significant reaction, close vital signs, being able to provide care if there was, you know, a reaction such as anaphylaxis. So I think those processes are getting better worked out now, and I think we'll see more access to the medication, and hopefully people who qualify and it may benefit are going to be able to receive it based upon what we best know with the data right now. And that was what guided our criteria for use.
Thank you so much, Dr. Costello, for sharing that with the group. So we got another question in asking, is convalescent serum still being used clinically? The literature seems to report limited benefit.
Yeah, so I mean, that's along with a lot of the therapeutics we've seen that have come out with COVID. As we use it more and see the results more, we change our stance on it. I can say that, you know, the problem with convalescent plasma or convalescent serum is unless you know what level of antibody titer is in that plasma, you're not really sure what you're giving that patient. I mean, is it really beneficial to that patient? The difference with these monoclonal antibodies that we know exactly that is a neutralizing antibody that will target that virus. And so I can speak specifically for our own institution, we've kind of gone away from using it. We were using it quite frequently in our hospitalized patients, but for right now, we've not really been recommending it as much. There's also some safety concerns that have come out with the convalescent plasma about potentially leading to increased thrombotic events or thrombogenesis happening. So I think it's a theoretical product that, when we look at the science, we say, oh, that does make sense. But in a real world practice, unless we know what type of antibody titer it is, it's not that beneficial, which has been the case with some other viruses that we've tried to use convalescent plasma with in the past.
Great, thank you so much. Any other hub members who wanted to comment on that before the next question? All right, so the next question we got was asking, can patients qualify for treatment with a rapid antigen test, or is a PCR test required?
Dr. Reese, I can take this one if you want.
Please do. Yes.
Okay, so so for the state criteria, it has to be by PCR. Although the EUA does discuss use of rapid antigen testing, even within our state, if say a patient in a nursing home has a positive rapid antigen that's confirmed via PCR. So at least our pandemic leadership, including Dr. Ayn Amjad, Dr. Clay Marsh, as well as other members of the pandemic leadership, Dr. Bill Ramsey, have continued to feel that PCR should be the confirmatory test that's utilized for, be based on the criteria used to give the monoclonal antibody. So at this time, it's solely a PCR-based test that needs to be positive in order to qualify for the antibody. So if there was a positive rapid antigen test, getting that confirmatory PCR in a patient in which you felt potentially could benefit from this therapy is what I would recommend in order to confirm that it was a true positive.
I would just like to say that hopefully, and Becky may want to talk about a number of COVID trials, clinical trials we anticipate opening in the next couple of weeks, but there is one on the monoclonal antibody among mild to moderate outpatients. And actually that NIH trial is going to use as inclusion criteria the rapid antigen test. And I think that makes sense because it is quicker. And as Becky mentioned, the earlier probably the better. And, there are, obviously false positives with every test. The real problems with antigen tests have been more that there have been false negatives, which is why if you use an antigen test on a symptomatic patient, you highly anticipate it's negative, you go ahead and get the PCR. In my opinion, and it's only my personal opinion, but I think, with the inclusion criteria of the NIH trial, there may be, or hopefully will be, evolution of use of the rapid antigen tests. I don't know if Dr. Sawari or Becky or others would like to comment as well.
Yeah, I mean, I think this was a discussion. So among the group yesterday, this was debated back and forth. Given that we know that the delay in timeline for the PCR in some cases and the benefit of getting treatment early. So yeah, I don't know that will evolve necessarily in a timely way, but I agree with you on the antigen test, that if it's a symptomatic patient with a positive antigen test, I think they would meet criteria. If we did not have a limited state supply that follows the state guidance.
I think that's well stated, Dr. Hodder, and I think we'll have to have ongoing looking at that. We've had this discussion really since remdesivir even, and so I think as more confidence grows in the antibody testing, in the feeling of the pandemic leadership of our state, and we have more data that comes back, I think we may see a shift towards accepting that as a positive treatment as well. And it's certainly something I think we'll probably bring up every time our work group meets, because it has been, even when we still had a remdesivir protocol.
Thank you all so much. We don't have any further questions at this time, so I didn't know if
I could ask a question, and I'm sorry to put Becky and Dr. Hodder on the spot, but as we start to have a vaccine come, I think I know the answer to this question, but you know, what's the thought of, I know at least I was on a call before about, you know, what's the concept of if you get monoclonal antibody and when you could get a vaccine, or, you know, if you get a vaccine and you still develop COVID, although the data looks really good in the vaccines, you know, would you then also be eligible for monoclonal antibody treatment? I think the answer is we don't know, but I would love to hear your thoughts on that.
Lisa, I've shared my thoughts, I think, earlier about this as well, that we don't know. There was one question that came up was, if I'm in a house with someone who has COVID and I don't know yet if I'm infected, could I get the vaccine? Because technically I'm, you know, under quarantine or under watch. And right now I think the guidance would be, well, until we know whether you're infected or not, we would not give you the vaccine because we don't actually know what happens in those people. But I would defer to Dr. Hodder, Dr. Serari for their thoughts on that as well.
RF, do you want to comment?
You gave me the harder question.
Well, of course.
It's, I think there is so much more that we are still continuing to learn about this. Short answer is we really don't know the answer there. I think these are going to be interesting conversations and challenges. And Becky, thank you for doing this. I am struck by how small the sample sizes are in terms of our EUAs. As you were speaking, what was running through my mind was, you know, there is some literature talking about COVID antibodies being thrombogenic. What's the monoclonal antibodies? Are they going to give me a DVT in 6 weeks, 8 weeks? You know, I don't know the answer to that. So just like the convalescent plasma has a signal of thrombogenicity, what is the signal for monoclonal antibodies? So we have certainly rushed carefully towards therapies that will hopefully turn the tide from an extremely high mortality disease among the greater than 65. But I think the concern about toxicity should not go away. I think we should continue to watch that signal carefully to see what to do. Dr. Costello mentioned Dr. Krupika's comments about using the monoclonal antibodies in the nursing homes, and I honestly think that's the place. We know that once the virus gets into our nursing homes, it spreads very rapidly. We go from 2 patients to 50 patients within a week, 10 days time, despite everything that we do. And then it is in that population that you see mortality rates going up to 10 to 15%. So those are just some broad comments. Again, definitely misdirected you to a different answer, you can see.
No, I think, you know, your sort of answer is very good. And one of the concerns has been with all of these EUAs that really the appropriate trials and understanding what populations these drugs work in, you cannot look very clearly at subpopulations with small numbers. And also to understand the side effects has been problematic. I mentioned a monoclonal study that we hope will be coming, and actually I had a long discussion with NIH, and that trial right now is giving Bamlanivimab open label, but will, with the next monoclonal, go back to placebo controlled for exactly the reasons that Dr. Sawari mentioned. And so I think that, you know, really we have gotten into a situation here, as, you know, we've seen with hydroxychloroquine, that everybody rushed to it. There was an NIH trial that maybe showed some impact, but in my opinion, you know, we're really sort of, because this has been such a dire situation, really sort of losing much of the rigor that we really need around the science. And I think that everybody always has to keep that in mind as these EUAs come through. I don't know, Becky, do you want to sort of just maybe summarize some of the other studies we anticipate opening? Becky is PI for 2 of them.
Mm-hmm. Yeah, so on that same kind of wavelength, trying to, even though we don't have identified yet these drugs that might work, there's 2 trials that I'm PI for the NIH. One is what's called the Active-5, and that's the big effect trial. And the goal of that trial is actually across multiple sites is to start comparing new agents or repurposed agents that might be beneficial in COVID-19 to look for that small effect that might develop on these small numbers. And if that looks good, then you push it forward into a bigger trial. And so that's one that we are hopefully going to, I'm hoping in the next 2 weeks be opening up, if not sooner. They're really trying to recruit to that. The Active-1 trial is another one that we are the PI for, that where I'm looking at another immune modulators. So this is not just monoclonal antibodies, but other immune modulators. So for instance, one of the drugs they were looking at early on was looking at not Humira, but one of the other agents to see if that was beneficial in terms of a kind of a cytokine control. And so those 2 trials are ones that are, again, across multiple sites, looking for small effects that develop to then progress it forward. I think in addition to that, we have a couple of anticoagulation trials that I'm not the PI for, but looking at both outpatient and inpatient benefits versus different anticoagulation therapies. And I think that's the main ones. Am I missing anything, Dr. Hodder?
Yeah, no, great job. Actually, Dr. Sharma is going to be the PI for what's called Active-4A, and that looks at various anticoagulation regimens for inpatients with serious illness. We have an outpatient Active-4B. Bill Lewis out in the Eastern Panhandle is going to be PI. And that one, we're not only going to open in Morgantown, but have some of our practice-based research network sites throughout the state. And then NIH is also working on 4C, the convalescent anticoagulation, which is going to be anticoagulation 28 days after hospital discharge. That protocol has not been finalized, but we're hoping to bring that here as well.
Great, thank you all so much. Were there any other comments or thoughts that the Hub members wanted to share with the group today? We don't have any further questions in the chat coming through as of yet.
Well, I think this is our last COVID echo of the year, if I'm correct. And I just want to really say thank you to we have one more, okay.
One more.
All right, well, good. Then I'll say my thank you then. But the team has been working really hard. There's now also an echo with nursing home residents that I think are nursing home centers that just kicked off this week. And so I think for those of you who practice in a nursing home as well, spreading the word about the importance of echo, I think we continue to learn from each other, including Dr. Reese's great presentation. So I wanted to say thanks to the echo team for all their work, because I really think it's helpful as we all continue to build the ship as we sail.
Thank you so much for the kind words, Dr. Costello, and we're grateful for all of you as well. Let's see, I think we actually did get one more question in, which is great. We have some time for that. So one of the questions was, can we be confident of the scientific rigor in the vaccine trials given the rapidity of the development?
I'll start out, but I think we can. I think, something that, despite the public push and the kind of pressure that has been put on getting these vaccines out, I think, from the FDA, they are really taking it to that scientific rigor that we need for those vaccines. The fact that the numbers look so good for it, but we're still 2-3 weeks since those numbers came out and not yet approved, I think they're doing their due diligence. I think the other part of it with, both the front runners is that the platform for that vaccine, that mRNA platform, is a newer platform that at least we know will not be infectious. So there's not a risk of giving infection through that vaccine, given that it's not a live or an active viral vaccine. But anyone else want to, Dr. Hodder, go ahead.
Well, no, I agree with everything you said. I think the other thing that's important to realize is you have a couple of other review panels. Number one, you have a data safety and monitoring board comprised of experts who really take very seriously, looking at these trials. These are not people who are employed by the manufacturers. These by and large are not government people. These are independent experts. And they really sort of look at the data, look at safety signals ongoing, And so I think that that is reassuring that you have their eyes. And also you have the ACIP, the CDC Committee on Immunization Practices, which, you know, came out with their recommendations, but they also look very, very carefully at the data. And these people are some of the most knowledgeable vaccine people you will ever find anywhere in the universe. And I think that that is very reassuring. And then finally, all of the companies, I think, were very concerned about, you know, whether people would have confidence, and also early on, you know, came out with a co-signed statement about, you know, the importance and integrity of having transparent data. So I think that all of those things are very reassuring. You know, you're never going to sort of, it doesn't matter what vaccine or what drug you approve, there may be a very, very, very rare side effect that you will never detect until you get it out in millions and millions of people. That's happened with drugs, you know, so there's no such thing here as saying zero risk. But what I always say, we know the risks of this virus and we probably don't even know all of them. And you're going to sort of, you know, when people call me about, you know, I'm not going to get this vaccine and this is why. And I say, so you're going to trust the virus with what we know and even we don't know for a, you know, sort of probably not zero, but infinitesimally small risk if these vaccines are approved. And I think that there's really going to be an issue with having the public have confidence in the evaluations. You know, we have to really have the wide majority of people vaccinated to really get the sort of protection across the population. And I think that, you know, this has really got to be for folks, you know, that sit in institutions and look at these things. I think we really ought to get out there and really hear and be part of the discussion, because I think that is going to be the major issue, is whether people will have confidence. I don't know how others feel, but I'm really concerned about that.
Yeah, I would just add in, I echo, pun intended, what Dr. Rees and Dr. Hodder said, but I think that we should and trust the process. I think the development steps that have made this be quicker is that, you know, we started producing vaccines simultaneously. We tried to start overlapping some of the things that I think typically take the longer, but the scientific process to evaluate the actual product has gone through the steps it would go through. And I think that we should trust that and be communicating that through our channels, because I do agree strongly with Dr. Hodder. We are actually working with a messaging team through the Reed College of Media, and we're doing looking at what are people's perceptions and understanding of the vaccine across sectors. We're doing both quantitative and qualitative analysis to try to get some true answers as to what is the hesitancy and what are their concerns, so that we as a health community can then provide scientifically sound information to them that address those concerns based upon what we know. And so I do think, you know, one of my mantras with vaccines are safe, vaccines are effective, vaccines save lives. I'm changing that to vaccines are safe, vaccines are effective, vaccination saves lives. Because if we have a vaccine that sits on the shelf and doesn't get the uptake, we're not going to get the benefits that we need to see, you know, real movement in this pandemic, so that we can all kind of get back to more typical interactions. Our economy can get back to where it needed to be. So it is very much, you know, our responsibility as a community to emphasize that the process that this has gone through, we should learn it and ask questions so that we understand it, but we need to be able to effectively communicate that to our patients, because we're going to be the first people to get it. We as a general-ish, you know, grouping as healthcare professionals, and then we're going to be recommending it to our patients. So I think we need to, you know, certainly have the information to feel confident in it ourselves, but then also to be able to relay that to the individuals who are going to ask us for our opinion, because that's going to be really important as we move forward. And we already knew that vaccine hesitancy was a concern amongst, you know, in the world. It was a global threat by WHO. So we certainly are going to need to have information that we can provide that's based upon science. And I think the science when it comes to the vaccine development can be trusted. We've seen trials be stopped when there's been a concern and that we've had to, look further into it. So it is following the scientific process with rigor, and I think we need to make sure that we're communicating that. And I mean, Britain already approved the Pfizer vaccine, and I think here in the United States, we're still sticking to our process so that it goes through the review panels to ensure that we're adequately looking at the data and ensuring that the recommendations, if it is authorized, and then the recommendations for use are based upon what the experts and the data and the science tells us.
Absolutely. Thank you all so much. And we got a chat in too saying that this is so important for people to hear. So thank you all so much. Before we do the poll at the end of today's session, any other questions from the participants or comments from the hub members?
I would just like to thank Dr. Reese for her terrific presentation, and thank Dr. Costello and Dr. Sawari as well for their input, which is really valued, and thank you so much. And all of you for giving an hour of your day.
Thank you, Doctor. And thank you, Dr. Hodder, for joining us and for everything you do as well. So thank you so much, Dr. Reese, for joining us today. I'm so happy we were able to find a time for you to join us. It's been wonderful, and we really appreciate your time. Thank you all for joining. So our next session will be on December 17th from 12:15 to 1:15. And that one will, I can't believe I'm saying this, be the last one of 2020. But we will be going into 2021 and continuing these COVID sessions. So just keep an eye out for reminders and everything. So I will quickly launch the poll. Anytime you finish the poll, just feel free to sign off. Thank you all so much. Take care, Beth. Thank you for joining.